When a firm is approaching clinical manufacture or first-in-human manufacture, there's the issue of stability. Is my product stable enough over the length of the manufacturing process, the filling and finishing process, to be stable when it's then used as a therapeutic molecule? If you know your product has stability limits, you have three options: reformulation, freezing, and lyophilization. My name is David Brett, and I'm part of the product service management team. And we give advice, especially to biotech and small pharma firms, on what they need to start their clinical manufacture. Most biotech and pharma firms will have some data on their drug product, because they've gone through preclinical research and now they want to start in a human trial. At that stage, they'll be coming to a clinical manufacturer and saying, okay, what requirements do I need for that manufacture? Firms coming from a preclinical stage will have some information on stability. They'll know that in a liquid formulation, it's stable for a number of hours or days. But they'll need to consider what that means for the clinical manufacturing stage. When you scale up to do batches that you require for the first in-human tests, so phase 1 and phase 2, you're going to have to scale both with the drug substance and drug product manufacturer and also the fill and finish. You may then be told you required longer times for the clinical manufacture than you had in the initial pre-clinical phase, and so then you have to consider your stability strategy. Do you need to go further with stability programs to keep the product stable during the manufacture? There's a number of issues to consider in a stability program. Of course, the short-term issue is my product stable enough to take it through clinical manufacture? But there's certain occasions where firms already know that they require a stability program. There are certain molecules that are already in the market, or very similar molecules, biosimilars for example, or classes of molecule that are already on the market in commercial production, where you know your molecule is very similar and you know it's going to need some sort of stability program. One are biologics, proteinaceous, antibodies and peptides, and the other group are things like lipids, lipid micelles, vectors for carrying vaccines that we see today in the COVID story. They also require stability at room temperature. So there's basically three options you have when you realize you have a stability issue. You can try and reformulate. You can freeze, just normal freezing down where the liquid formulation becomes solid. Or you can lyophilize, which is to make a freeze-dried powder. Each one of them has advantages and disadvantages. Formulation obviously would be the gold standard if you could do it. It'd be great to have a liquid formulation that works and stable. You'll have a formulation to begin with, and it'll have to have been formulated as a drug product with excipients to be able to be injected into animals, into mammals. So it has some degree of stability. But if you scale up and you want to go into humans, and the manufacturing process is then obviously longer, you need a greater degree of stability. So you may have to look at different formulation options and try and reformulate to get that stability. Maybe a change in excipients, maybe a change in concentration. Most firms will do the reformulation if they can, and they'll do it probably in parallel to either freezing or lyophilization. But there's no guarantee that attempting a reformulation will work. You may try several candidate new formulations and they may not be any more stable than your original one. Sometimes, in a large number of cases, it doesn't work, and so you have to consider the other two options. In the majority of cases, if you're approaching first in human, then you may look to freeze your product to get round the stability issue. Freezing is cheap and easy, gets you started, and a lot of firms in early phase will just freeze their product to get started, to have a stable product they can work with. But that obviously has issues with it as well. I mean, one issue, of course, is your product may not hold up to being frozen. Freeze-thaw cycles can damage proteinaceous compounds, and so you have to test that first. So a freeze-thaw cycle at a number of occasions during the manufacture, the compounding, the filling, the refreezing, the visual inspection, all may require then freezing steps. So there might be multiple freezing, thawing steps along the manufacture process. Of course, the second issue to think of is a longer term issue, which is, do I want to be stuck with a more complex and more expensive cold chain issue later on in commercialization and development? If there are markets you're moving into where you know your product will be supplied to those markets where their cold chain is not a given, some countries don't have the capabilities to keep things along the whole chain cold, then you may want to look at lyophilization. Lyophilization is a process of freeze drying. That means using cool temperatures and a vacuum to remove the residual moisture from your compound, and so it becomes a frozen powder, and then you increase the temperature up so it becomes a stable powder at room temperature. Freeze drying is probably something most firms won't approach straight away, primarily because it's time and expense that adds to your project when you want to get started and get your results in phase 1 or 2. So if you require lyophilization as a step, it's not something you do lightly. You have to develop a lyophilization cycle to fit your molecule. There isn't a standard process that you can just apply, so it's a different development cycle that you have to take into account. That means that you're possibly going to spend 12 to 18 months developing a robust commercial lyophilization scaled process. If you realize there is a issue of stability and you're going to require a longer term stability strategy, which may include lyophilization, FETA obviously would recommend to start early because the lyophilization cycle development process may require several formulations. Starting early in the process gives you an advantage when you're starting getting your clinical results. Considering a longer-term stability program, a lot of firms will then look at their considered global market. You know, are they going to be supplying to many different countries in many different scenarios? If that's the case, and it becomes a widely distributed drug product, maybe a blockbuster, they may need to consider the lyophilization because it's going into all sorts of hospital, outpatient, and rural settings maybe, where the cold chain isn't a given. But obviously when you have positive data and you want to move maybe into phase 2 and phase 3, then you have to look at the longer term. Do you want to stay with a frozen product? Do you want that complexity of the cold chain in phase 3 and commercial supply? Or do you want to say, okay, now is my time to look at developing a lyo cycle so that I have long-term stability and I can provide commercial supply to different markets? You can't put all your eggs in one basket in that sense. You can't just go for reformulation. You have to look in parallel at either freezing or lyophilization. Each of those options, reformulation, freezing, lyophilization, has its advantages and disadvantages. You can always try and reformulate, but there's no guarantee that your reformulation work will actually provide you with a better or more optimal, more stable product. So freezing is great if your molecule can stand the freeze-thaw process. It's cheap, it requires very little development, or if that is not applicable to your molecule, or you don't want to stay with freezing because it's adding a more complex cold chain that then would require your distribution network to have freezers, you can look at lyophilization. If you want to license your product, the licensing agreement will be discussed based on the effectiveness of your molecule, obviously, but also how much effort and work the pharma firm has to do to get it to phase 3 in clinical supply. So if you've done some of the work already, you've done lyocycle development for stability, it adds to the value of your product. You can, with that investment in time and effort, you can pass that increase in quality and commitment to your product on to other licensees, for example, if you're thinking about licensing your product later. But of course, time and money is, especially in the early phase when you don't know your molecule is going to be either effective or competitive, is something that you don't want to consider straight away. But as the process and your trials move on, of course, you then have to consider the long-term impact. Your approach to stabilizing your product can have a big impact on its market profile.